Okay, so welcome back to this next video in which we are discussing desmosomes. Okay, right, so we're in the process of discussing the structure of all of the desmosomal catarins. So we've seen that there are 7 different genes for desmosomal catarins, okay, which are divided into these 2 major families, desmoglines and desmocolins. Okay, so there are 4 different genes for desmoglines, okay, whereas there are 3 different genes for desmocolins. However, we overall have more desmocolin proteins because each of these 3 genes for desmocolin proteins codes for 2 different splice variants, the desmocolin A splice variant and the desmocolin B splice variant. And this is the case for both, all of desmocolin 1, desmocolin 2, and desmocolin 3, meaning that overall we have 6 different desmocolin proteins. Okay, right. However, what's lovely about this is all of the desmosomal catarins have the same basic structure, which we're going to discuss now. Okay, right. So, firstly, they are all individual polypeptides, okay, and they are integral membrane proteins which span the membrane a single time. Okay, so I'm going to draw this now. Okay, so they're going to have a membrane spanning alpha helix. Now I haven't actually drawn it as an alpha helix here. Okay, people generally don't, but this will be an alpha helix here that is spanning the membrane. Okay, so they have this single membrane spanning alpha helix here. Okay, and I'll highlight that in red. Okay, right. Now, the extracellular domain then of our desmosomal catarin is going to have 5 really important domains. Okay, so the first one is known as the extracellular anchor. Okay, the EA domain. Okay, then following that we're going to have these 4, I didn't tell you the actual number we're going to have, but we're going to have extracellular catarin domains. Remember, these domains that are utterly essential for any catarin protein to have, and we're actually going to have 4 in the case of all the desmosomal catarins. So let's draw these now. So here's 1, here's another one, okay, here's a third one, and then we've got a 4th one. Okay, right, so let's colour in these extracellular catarin domains in green. Okay, and then on its own right at the end we've got an extracellular anchor, which is that EA domain here. Okay, now remember the extracellular catarin domains, these are all around 110 amino acids, and they all are going to have pretty much the same structure, very similar structures. Okay, right. Then we have the amino terminus of the polypeptide right at the end here. Okay, so this is our amino terminus of the polypeptide, and that means since we're only going to span the membrane once, the C terminus of the polypeptide is going to be intracellularly. Okay, so we have a short cytoplasmic tail, okay, and then we're going to have the C terminus intracellularly there. Okay, so let me just put a little bit more colour here. We have our extracellular anchor here in vivid purple. Okay, right, so I'll just label these portions up. So the EA domain stands for the extracellular anchor domain. Extracellular anchor. And the ECs, remember, those stand for extracellular catarin domains. So we have 4 of these extracellular catarin domains. Okay, so these are all extracellular catarin domains. Okay, right, so now what we want to discuss is firstly what the difference between the desmocolin A splice variants and the desmocolin B splice variants is. We then want to discuss how these desmosomal catarins on one cell are going to interact with a desmosomal catarin on the other cell. So we'll want to draw another cell here and show the interaction between 2 desmosomal catarins. Okay, but firstly, how do the 2 splice variants of the desmocolins differ? Okay, so remember we have these 3 genes for desmocolins, desmocolin 1, desmocolin 2, desmocolin 3, and all 3 of these genes have 2 splice variants, an A variant and a B variant. Now the A variant is longer than the B variant. Okay, so you could call the A variant the long variant and the B variant the short variant. Now, where are these 2 variants longer or shorter? Okay, so basically the difference is in the cytoplasmic tail. Okay, the A variants, which I'll have in purple, they have a slightly longer cytoplasmic tail than the B variants. Okay, so to make this more transparent, let me draw a little picture here. Okay, so now I'll just represent a desmosomal cadherin as a line, like so. So this is the extracellular domain, the membrane-spanning alpha helix, and the cytoplasmic domain. Okay, so let's say that this is an A splice variant of a desmocolin. Okay, this would be a B splice variant. It's got a much shorter cytoplasmic tail, basically, but the extracellular domain, which is responsible for binding to another desmosomal cadherin, is basically going to be unchanged between the two splice variants of the desmocolin. Okay, right, so that's the difference between the A and the B splice variants. I should probably just label them up. So this is the A, the long one, and this is the B, the short one. Okay, so here is A, and here is B. Okay, right. Now what we want to talk about is how two desmosomal cadherins on opposite cells are going to interact with one another. So over here, remember this is cell 1 that we've got here. Okay, and now we're going to try and draw on cell 2 over here. Okay, so the desmosomal cadherin on cell 2 is probably going to have to be a little bit more squashed to get everything to fit in. So basically the first thing to say is that the way the two desmosomal cadherins bind to each other is that the N-terminal extracellular cadherin domains of the two desmosomal cadherins are going to bind. Okay, so this final extracellular cadherin domain, or depending on the way you look at it, the first one, because usually we go from the amino terminus to the C terminus, so this is the first extracellular cadherin domain here, or you could call it the N-terminal extracellular cadherin domain. This is the one that's going to be involved in binding to the other desmosomal cadherin on the other cell. Again, it's going to bind to the N-terminal extracellular cadherin domain on the other desmosomal cadherin on the other cell. Okay, so here's the amino terminus of the second desmosomal cadherin, and this is an extracellular cadherin domain. Okay, so I'll colour this in green here. Okay, then let's finish this structure off then. So we're now going to have 3 more extracellular cadherin domains, which I'm going to squash in a little bit now. So here's the 3rd one, here's the 4th one here, okay, and then we have our membrane here, our membrane spanning alpha helix, like so, okay, and then our C terminus here. Okay, whoops, and what have I forgotten to put on there? I've forgotten the extracellular anchor. How am I going to repair the damage? Okay, so we'll have to turn this into an extracellular anchor. Then we'll put the cell membrane here. Okay, right, so I'll try and hide this. So here in purple, this is the extracellular anchor. Okay, ignore these portions, those should not be there. Okay, and now we've got the other 3 extracellular cadherin domains, which I'll show in green here. Okay, right, so there is our desmosomal cadherin on the other cell. Okay, right, so this is how you get this interaction between a desmosomal cadherin, or a cell adhesion molecule, for that's what this is. It is fundamentally in the massive great category of cell adhesion molecules, interacting directly with another cell adhesion molecule on cell 2 over here. So this is our second cell. Okay, right, so what I now want to talk about is the concept of homophilic and heterophilic binding. Okay, so let's now talk about homophilic versus heterophilic binding, because when we're talking about desmosomal cadherins, heterophilic binding is actually going to be extremely important. Okay, so we'll start with what homophilic binding means. Okay, so basically, homophilic binding refers to when the first desmosomal cadherin here on the first cell is the same type as the second desmosomal cadherin that you have here on the second cell. Okay, so basically, they both need to be desmoglein, or they both need to be desmocolin. Okay, so number 1 will be a desmoglein desmosomal cadherin, and number 2 will be a desmoglein desmosomal cadherin, or number 1 will be a desmocolin desmosomal cadherin, and then number 2 will be a desmocolin desmosomal cadherin. Okay, so homophilic binding doesn't actually mean that the two proteins need to be identical. It doesn't mean that if this is a desmoglein 1, this also has to be a desmoglein 1. It's not quite that strict. What it means is that if this is desmoglein 1, this has to be also a desmoglein desmosomal cadherin. But it could be desmoglein 2 or desmoglein 3 or desmoglein 4. Okay, just as long as it's in the same family. Okay, we're not quite strict enough to insist that it has to be the identical protein. Okay, so remember, homo means same, just in case you're wondering. So these are interactions between two cell adhesion molecules which are the same, or two desmosomal cadherins that are the same, but it doesn't quite have to be an identical protein. It just means it has to be in the same family, basically. Okay, so if it's a desmoglein here, this has to be a desmoglein here. If this is a desmocolin, this has to be a desmocolin. Contrast that to heterophilic binding. Okay, and heterophilic binding is believed to be more important in the desmosomal cadherins than homophilic binding. Okay, which is rare. If you look at the classical cadherins, for instance, they much prefer homophilic interactions than heterophilic interactions. Okay, right. So in heterophilic interactions, what's going to happen is number 1 might be a desmoglein, okay, and then number 2 will be a desmocolin. Okay, or it could be the other way around. This could be a desmocolin and this could be a desmoglein. Okay, so to give an example, we could have desmoglein 2 here, okay, bound to desmocolin 1A, let's say here. Okay, and that would be a heterophilic interaction because the two desmosomal cadherins that you are binding together in this way are from different families. Okay, so hetero means different, so this means that the bond is between two desmosomal cadherins that are from different families and is therefore a heterophilic bond. Okay, right, so that's the binding of two desmosomal cadherins together. Okay, now what we want to discuss is what the cytoplasmic tail here is going to bind to. Okay, so firstly let's give the motivation for this. So what have we found so far? What have we done? We have put a nice little desmosomal cadherin in our cell membrane, and we have attached it to a desmosomal cadherin in the other cell membrane. Now the question is that going to attach the cells together in any sort of structurally sound way? Okay, and I'll give you a clue, this is not how cells do it. This is not the complete picture yet, it's not finished. Okay, but to understand why, let us remember that the cell membrane is incredibly flexible. Okay, this is really, really easily distorted. Okay, so the cell membrane is not a rigid structure. So you've attached one non-rigid structure to another non-rigid structure. That's not great, basically. That's not a great attachment between the two cells. Okay, so really what we need to do is attach the rigid structure of two cells together. Now, what is the rigid structure of two cells? Well, let's draw a picture here. So this is our sort of like immature picture of a cell. Okay, this is the picture of a cell that we draw because it looks nice and it looks simple. Okay, but in reality, cells do not look like this. Okay, and that's because in reality, the cytoplasm is not just one confluent gloop, okay? It is full of a meshwork, okay? A meshwork of proteins. Now, if I was to actually draw this, it would look like so. And I'm going to do this in colour because if I do it in that black pen, it will just end up looking awful. Okay, so really what you've got inside the cytoplasm is just endless protein filaments, and there are mixtures of loads of different protein filaments that are here. Okay, and these protein filaments, they all interlink, and they form a huge, great meshwork that holds the cell up. These are the rigid structures of the cell. Okay, these hold things in their positions. This is the reason a cell has a consistent shape. If you just had the cell membrane, the cell shape would change all the time because it's just a flexible gloop, basically. Okay, so the fact that you have this rigid protein meshwork everywhere within the cytoplasm that is holding the cell in this shape, this is the rigid structure of the cell, basically, and this is what our desmosomal caterins need to attach to if they're going to make a viable connection between these two cells. Cell 1 has one of these rigid meshwork of proteins. Cell 2 has a rigid meshwork of proteins. If you want to connect cell 1 to cell 2 properly, you need to connect this rigid meshwork of cell 1 to the rigid meshwork of cell 2. Now, there's a nice name for this rigid meshwork that you have within cells, and it's known as the cytoskeleton. Okay, so basically we need to discuss how these desmosomal caterins are now going to attach to the cytoskeleton. Okay, right. So firstly, we're going to talk about which sort of protein that is part of the cytoskeleton are desmosomal caterins going to bind to. And this is really important because this is what distinguishes desmosomal caterins from the classical caterins. Okay? Basically, desmosomal caterins are going to bind to structures within the cytoplasm of cells known as intermediate filaments. Okay, so desmosomal caterins, they are going to bind to structures that are part of the cytoskeleton, which are known as, whoops, what am I doing? Intermediate, I'll turn that into a T. Okay, intermediate filaments. Okay, right. Now, contrast this to the classical caterins, which basically bind to actin filaments within the cytoskeleton. Okay, right. So I want to talk about what an intermediate filament actually is. Okay, but firstly, let me give you a name that you're probably more familiar with. Okay, so the main example of an intermediate filament, okay, that is present within the cytoskeleton of cells are keratin filaments. Okay, so keratin is the main example of an intermediate filament. Okay, it's the one that everyone has heard of. Okay, people who know nothing about biology have heard of keratin. Okay, it's what nails and hair is made out of. Okay, so we want to see what the generic structure of an intermediate filament is, and there are other intermediate filaments. They're less well known, they're less famous than keratin. Okay, but there are other ones that are components of the cytoskeleton as well. But we'll concentrate on keratin. Okay, but all intermediate filaments have the same basic structure, and I want to now tell you about this same basic structure now. Okay, so basically, the way that you build an intermediate filament is you have to start off with a monomer, okay, which is a single polypeptide. Okay, so we'll start off using our motivating example of keratin with a keratin monomer. Okay, now there is not just one monomer for keratin, there are a huge number. I think it's something like 52 genes in humans that are currently known to code for monomers of keratin. Okay, so there are a huge number of different monomers of keratin which all have different sequences of amino acids, okay, and can all assemble into keratin filaments, and they all mix together to make a filament that has loads of different monomers in. But the basic structure that they all have looks something like so. Okay, firstly they'll have their amino terminus, so I'll put it here. Okay, and then they are going to have a portion known as the rod domain, which is an alpha helix, basically. Okay, so let me show this. Now, an alpha helix, let's discuss alpha helices in a little bit more detail. An alpha helix is a spring-like structure. Okay, so if this line represents the polypeptide, okay, so it represents amino acid after amino acid after amino acid, then basically in an alpha helix what's going to happen is the polypeptide is going to fold round into a spring-like structure like so. So let me just guide you with this. At the moment we're at the back here, okay? Then we're coming forward out towards you, okay? Then we're going looping round to the back again, okay? We're coming round to the front again, okay, like so. Okay, so this is what's known as an alpha helix. Now, there's a subtlety here that takes a little bit of visualization capacity to understand. Okay, there are two different types of alpha helix. Okay, and the one that I have just drawn here, which is the type that we're going to actually have within a monomer of an intermediate filament or a monomer of keratin. Okay, this is what's known as a right-handed alpha helix. Okay, right. So, contrast that to a left-handed alpha helix, which I'll show you in a minute. So what's the characteristic that a right-handed alpha helix has to have? Well, basically, it is that as you loop round, okay, so let me talk you through this again. We're coming round to the front at the moment, then we're going back to the back, and we're looping round, coming out to the front again, and then looping round to the back. Which direction are we going round in as we go down the alpha helix? We are circling round in a clockwise direction. Look, we're coming round here, we're looping back and going in a clockwise direction. So as you come down from the top of the alpha helix to the bottom, you go round in a clockwise direction. Okay, now obviously if you're going from the bottom upwards, you will go in an anti-clockwise direction. Okay, like so. As you can see, I'm looping around in an anti-clockwise direction now. Okay, and you might think, but surely that's trivial, because if I just said that this was the top and this was the bottom, okay, so if I flip that round, surely it'll be the other way round, but you'd be wrong to say that. Look, if we flip it round, and now I work my way from the top to the bottom, okay, which direction am I going? It's a little bit harder to follow, but look at the direction that I'm going in now. Okay, we are looping round in a clockwise direction. Okay, so